Welcome to No Worries Biology. My name is Anja Doyle, and today I would like to talk about the replication of DNA. This video is meant as a summarizing review, so it's best suited for those of you who have already learned about the replication of DNA. When James Watson and Francis Crick worked on their three-dimensional model of DNA as a double helix in the 1950s, they realized that this structure would make it very easy for this macromolecule to be copied. DNA must be copied before the cell can divide, so that each of the daughter cells can receive a complete DNA set. This process of copying the DNA is called identical replication, or just replication of DNA. It takes place during interphase, and more specifically during the S phase of the interphase, because the S in S phase stands for synthesis, meaning something new gets built or assembled, and in this case it's DNA. So the identical replication of DNA ensures that each daughter cell possesses the same amount of DNA and a set of the information encoded within the DNA after cell division. We already know that the strands of the double helix of DNA are complementary, meaning that by looking at one half of the double helix, you can tell what the other half must look like. DNA replication works by following this exact same idea. The double helix gets separated into two single strands, and then each of these single strands is used as a template to create the missing half anew by following the principle of complementary base pairing. To make this easier to understand, let's have a closer look at what's actually going on during the replication process. Well, first of all, a complex of multiple replication enzymes attaches to a specific site on the DNA called the origin of replication. This is an important fact, but it's not shown in this sketch here, just to keep the sketch as simple as possible. In prokaryotic cells, there's just one origin of replication. In eukaryotic cells, there are actually multiple origins of replication. One of these replication enzymes, called helicase, starts by unwinding and unzipping the DNA, and it does so by breaking up the hydrogen bonds between the base pairs. Another enzyme called DNA polymerase takes individual nucleotides which carry the correct complementary bases and attaches them to the nucleotides in the template strand. The sugar and phosphate group molecules of these individual nucleotides form bonds with each other, and by doing so they create the new second backbone of the DNA. In prokaryotic cells, this happens at a rate of about 500 nucleotides per second, while in eukaryotic cells the rate is about 50 nucleotides per second. As a side note, in eukaryotic cells, there's actually not just one type of DNA polymerase, but three. But that, quite honestly, is not very important when you just want to understand the main principle of DNA replication. So we're going to neglect that here. But keep it in mind in case that is important for your biology lessons. So the basic mechanism of DNA replication is complementary base pairing. But of course, there are some specifics that make the whole issue a little bit more complex. Let's look at them and dive a bit deeper into DNA replication. While DNA polymerase is capable of elongating a DNA strand, it actually cannot start a new one by itself. Therefore, it always needs a specific starting point. Let me use an analogy here. Maybe as a child, you learned how to crochet or how to knit. Well, I did anyway. And once everything was underway and set up, it actually wasn't too hard to conduct these repetitive motions. But when I was little, I was never able to start a new piece by myself. I always needed my mom to start it and to set everything up, and then I could take over. Well, that is kind of how it is with DNA polymerase. Once the beginning is made, it takes over. And because DNA polymerase doesn't have a mom, the starting point is created by an enzyme called primase. And the starting point itself is called a primer. To keep things simple, in our sketch here, the primer is shown as a star. In cells, of course, primers are not star-shaped. They actually consist of a few nucleotides of another type of nucleic acid called RNA. Another fact to keep in mind is that DNA polymerase can only work in one direction. It elongates the DNA strand in 5', 3' direction. That means that at one of our template strands, DNA polymerase can simply follow helicase as it unwinds and unzips the DNA. On the other strand, however, DNA polymerase has to work against the direction of helicase. Therefore, at this template strand, the DNA has to be synthesized bit by bit. After helicase has unzipped a small portion of the DNA, a primer is created so that DNA polymerase can start working. Meanwhile, helicase continues to unzip the DNA, so that a little bit further along, a new primer can be created and another DNA polymerase molecule can start working, and as we already know, it synthesizes the new strand by following the principle of complementary base pairing, and it does so until it reaches the previous primer where it stops working. Meanwhile, helicase still continues to unzip the DNA, so a little bit further along, another primer can be created, another DNA polymerase can start working, and it does so until it reaches the previous primer. We have seen that one of the strands is replicated continuously and the other strand is replicated discontinuously. However, both of these processes happen at the same time. Let's have a quick look at how both of these processes happen at the same time. And because the continuous replication is just a little bit faster because it can just follow helicase, and the discontinuous replication is just a little bit slower because it has to wait a little bit, the continuously replicated strand is called the leading strand and the discontinuously replicated strand is called the lagging strand. The fragments in which the lagging strand is created are called Okazaki fragments. They are named after Reiji Okazaki and Tsuneko Okazaki, who discovered the discontinuous nature of the replication of the lagging strand in the 1960s. In prokaryotic cells, Okazaki fragments are about 1,000 to 2,000 nucleotides long, and in eukaryotic cells, they are only 100 to 200 nucleotides long. Finally, and as a last step, the RNA primers that were used in the leading and lagging strand are replaced with DNA nucleotides by the enzyme DNA polymerase. And the sugar phosphate backbone of these individual nucleotides gets connected with each other and with the rest of the newly created DNA strand by an enzyme called ligase. The result are two identical strands of DNA that consist of half the mother strand and half a newly created strand. And this is why the mechanism of DNA replication is said to be semi-conservative, meaning half of it is old and half of it is new. This is it for today. I hope this video might have been of some help to review the process of DNA replication. On www.noworriesbiology.com you can find lots of materials on genetics or other topics. So just look around and see if you can find anything that helps you or that interests you. I hope to see you soon.